time. I'm moving right to Martin-Immanuel Bittner from Arctoris, United Kingdom. He's going to talk about implementing the fully automated kinase inhibitor characterization for artificial intelligence-driven drug discovery using a robotic system. Martin, the floor is yours. Thank you so much.
Wonderful. Thank you very much for these very kind words. I'm just going to very briefly share my screen. You should be able to see my slides.
Yes, we can see it. Perfect. Thank you.
Wonderful. Thank you very much. Well, it's wonderful to be here. Thank you very much for having me as a speaker at this event. What I really want to focus on is the topic of where I think very similar to the talk we just heard, where technology really meets biology in what I think is a very new and a very exciting way. But before we go into the talk itself, I would like to very briefly start by saying that what I think motivates all of us here is that we do really need new and better therapies. And that is something which I kind of very firmly believe in, kind of as a clinician by background, that we have to do everything we can to find these new therapies and to find ways to find them faster and to find them in a more efficient way. The reason why we need to work on the efficiency is because the drug discovery process is, as we all know, extremely long, very expensive, and fraught with challenges, lack of reproducibility, lack of success, etc. Just looking at the success rates that we have when moving a particular program from one stage to another. So just looking at the current success rates, we can already see that there is quite a sharp drop over time. And if we compound it, we arrive kind of at these rather dismal success rates that are plaguing the industry. What it also means is that the cost of bringing this one single new drug to the market is extremely high. And that is something that we must take into consideration, and that must also be an inspiration for us to try to use technology, be it AI or be it robotics, to do things differently. This slide kind of shows Irum's law, kind of the way it has been termed in analogy to Moore's law, to show how the number of approvals over time has been going down, or respectively, how the cost per drug has been rising. So in other words, the amount of money it takes to bring a single new drug to market, to go through all the regulatory stages, has been rising constantly. And that is something which is a huge concern, because that means at some point there's going to be a situation where the sustainability of the R&D model we believe in is no longer to be a given. What have we identified as some of the key issues and some of the key bottlenecks? The first one is data quality. We know that most of the data used in biomedical research is poorly annotated, unstructured, and oftentimes unreliable. This evidence comes from mostly pharma companies who try to reproduce findings from the literature and to only succeed in a very small percentage of cases, which is worrying. At the same time, we also see that there is a very inefficient use of our most valuable resources. Scientists, the scientists who are currently working in the labs, are the most valuable resource we have. Their creativity, their knowledge, their ingenuity is what drives projects forward. But at the same time, they spend so much of their work time with manual laboratory work, and only a fraction of their time with more intellectual tasks, such as keeping up with the literature, thinking about the next project, planning their experiments, etc. What that is linked to is the fact that laboratories really have not changed in many decades. All of us kind of walk into labs every single day, and I think we just take it for granted that laboratories look a specific way. But if we think about it, that same laboratory will have looked almost identical 20, 30, 50, even 60 years ago. Of course, by now we have kind of a PCR machine and there is a computer somewhere in the corner, but the work mode itself has really not changed. One of the core technologies in all industries, but I also think in particular within drug discovery, is artificial intelligence, which might hold the key to finding greater productivity and to seeing programs being developed for new targets with new, potentially kind of very, very rewarding applications. So those are approaches where AI can really come in as a very important part of our overall toolkit. These are just a few news pieces from the last few years that highlight what AI can be used for, how broadly it's being used, and who are kind of some of the major organizations in this space. There is in silico medicine, there are AstraZeneca and Berg, there's eTherapeutics, Schrödinger, of course, Exscientia. So those are kind of some of the big names, both kind of in the US, Europe, as well as in Asia Pacific, we're really working on new applications for AI, which can have a massive impact. So for example, there was a recent Nature Biotechnology paper published by Alex Zavaronkov from in silico medicine, demonstrating how they used AI to really kind of work on a new program in a far more efficient way compared to what has previously been thought possible. Interestingly, we can also see that this trend is further accelerating. If we track either the number of publications or the number of companies working on AI or machine learning for drug discovery, we can see a very, very sharp increase over time. There really is this increasing realization that AI can be an extremely powerful way of working on generative chemistry, target ID, and many other ways as well. One resource that I find particularly useful is one of the blogs curated by BenchSci. BenchSci itself is an AI-driven, very well recognized AI-driven company in the US. And what they kind of built over a long time was a curated list of different areas within which they spotted interesting, promising AI-driven companies that work somewhere within a drug discovery ecosystem. And as you can see, there's quite a long list, starting off from information synthesis, via biomarker establishment, data generation, repurposing, drug design, down towards clinical trials and real-world evidence. So we can really see that across the whole drug discovery process, across the whole biomedical research value chain, there are opportunities for AI to come in and for AI to really change the way we think about research and to change the way we approach research. And that is something which I find extremely promising, because we know that not all of these approaches will succeed. We know there will be failures and there will be setbacks, but we also know there is a huge breadth of different approaches, different groups in academia and in industry working on these very, very promising ideas. The one that I'm going to pick out is kind of what I very, very briefly kind of alluded to already, which is generative chemistry. In this case, a deep learning approach for de novo drug design for a kinase inhibitor in kind of a DNA damage response pathway, where there were some very, very interesting things happening that were, in this case, kind of done by Alex Zawaronkov's group, where we can see that the application of their model, Gentler, led to them progressing from choice of targets down to having several very, very interesting compounds synthesized in a very short amount of time. So as you can see on the timeline, this was a total of 46 days. And this is something which is a very, very interesting study that can really serve to demonstrate how effective machine learning can be, in this case, when being applied for the drug design aspect, especially when also taking into consideration that the study was recently being taken as the basis for another study that they've been publishing this year, which has been going one step further. We're also including AI for target identification in the first place. So in other words, it's also very interesting to think about how some of these AI technologies can be seen in almost like a Lego system, where you can take kind of different bricks and you can combine them to something which then yields even greater returns. So that's basically just kind of one study I wanted to pick out, which I found particularly interesting and insightful, to focus on the abilities that AI has. But at the same time, we also need to think about things that currently are holding the industry back. And one of the key things that are holding the industry back is access to data. So as we've seen at the very beginning, we do have issues with data, but we also have this huge promise of AI. And so what we need to think about is, how do we combine those two? How do we marry those two needs? One for greater deployment of AI models, and one for data to train and validate those models. In our opinion, the answer is robotics. If we again look at the drug discovery ecosystem, the drug discovery funnel, as of now, robotics for most people in the industry meant high throughput screening. That was more or less the definition of robotics within drug discovery. That people would have a huge facility that can screen 500,000 molecules, 1 million molecules, 5 million molecules. For example, in a biochemical assay system, where you would test all of your compounds against a particular target, and you would then find the hits that you then want to work on. This is kind of everything which, for the most part, robotics has been used for within drug discovery. But over the past few years, it has become possible to automate a far, far broader range of tasks. It has really been the past 5 to 10 years that have shown that even within target validation, within hit to lead, with lead optimization, partly even within the preclinical space, there are opportunities to use automation, to use robotics, to lead to higher quality data generated at larger scale. I think it's extremely important to point out that while the basic technologies are very similar, robotic arms, liquid handlers, this really has been quite an evolution that was driven by academic groups, that was driven by companies, that was driven by large corporations, where there was also kind of a lot of work going on standard creation. But where we are right now is in the very unique situation where we can really harness the power of robotics for a far broader range of tasks than it would have been possible even just 1 or 2 years ago. So this really is a development that has been going on for a few years, but has now been reaching a stage where it really is extremely interesting and something that is worth taking note of. One other measure or parameter is also to look at who else is working on this and what are some of the responses kind of we've seen in the industry. There is, for example, in Citro in the US, there's Recursion Pharmaceuticals, there's Ginkgo. So we can see that in many different ways, robotics has now become a topic that is of increasing interest for people in the industry, where several companies have now made huge investments in the space as well, because they were driven by the realization that especially when you want to rely on machine learning and AI, what you need to think about first is data quality. There is this saying kind of in the machine learning community, garbage in, garbage out, where the quality of the input data really determines what the quality of the models will be and what the usefulness will be. And so I think it's extremely important to bear in mind that something which works in recognizing cat videos on YouTube, that the very same principles also apply to drug discovery, where we really need to think very carefully about how we generate the right type of data that can then be used for these purposes. The reason why robotics is so transformative is because automation works on different levels. One is the cost and speed level, where suddenly running the same project can be completed with a smaller investment, both in terms of time as well as monetarily. And it also works on the quality angle, because we know that thanks to automation, data quality can be considerably improved. I'm going to go into further details on the following slides, but I think it is extremely important to really kind of see those two areas, cost, speed, efficiency on the one hand, and quality on the other as independent but closely related factors, both of which contribute to the great success of robotics and to the great promise it holds for the future. Digging a bit deeper into the question of data quality. So this is kind of using our company as an example, but it really kind of applies to automation when done the right way in a slightly broader sense as well, where we think about what we're currently doing and what we could be doing. What we're currently doing within drug discovery, within biomedical research, is looking at the tip of the iceberg. Currently, there are isolated data sets. There are very ambiguous methods and protocols. Storage systems are usually fragmented. In many cases, scientists will have USB sticks which contain some of their data when they walk around in the labs. And data formats are usually not standardized. Just to give an example on what some of these points will mean in real life. If we think about a scientific protocol, then normally kind of that will read along the lines of shake the sample, or it will read along the lines of shake the sample gently, or it might even be less specific, it might say mix the sample. In all of these instances, there is a lot of ambiguity on what that actually means and what the scientist is actually supposed to be doing. If we take the broadest scenario, mix the sample, that could be shaking, stirring, vortexing for half a minute, for 3 minutes. No one really knows. And we know that these differences in how a method and how a protocol is being carried out has downstream effects on what the results data will look like. For example, because cell-cell interaction has been impaired, or because cells might have been slightly damaged and will then show a different stress response, etc. At the very same time, non-standardized data formats also includes questions such as ontologies, where a lot of researchers, even within one research groups, will have very different ways of referring to their data, referring to their proteins of interest, etc. So it currently really is very, very difficult to pool data from different researchers. And that is something which can change. So when we look at the bottom part of the graph, where we see everything which is currently below the waterline, we can then see that thanks to automation, thanks to robotics, and thanks to kind of sensor arrays, we can suddenly have a full digital data capture, where we know exactly what went into every single experiment, where protocols are standardized, where, for example, mixing a sample is defined as 30 seconds at 22 degrees centigrade with 20 rotations per minute, for example. So it really is about bringing the benefits of standardization and of structure to this environment. What that enables us to do is enables us to have very comprehensive and transparent audit logs, audit trails, as well as feeding these various pieces of data into interoperable databases, which means a research group that might consist of 5 researchers, or a biotech company that might consist of 100 researchers, can use their data in a far more efficient way, because they can really bring together different data sets from different points in time, from different scientists, from different sites, because they know they will be inherently comparable. Another very important aspect is also reagent provenance. We know that in many laboratories around the world, cell lines and cell identification is a huge issue, where only a fraction of the cell lines currently used in research are actually well characterized, tested for identity and for contamination, and then used according to best practices. The latest numbers are that at least 1 in 2 cell lines will either be contaminated or misidentified. So in other words, we already have a massive issue when it comes to reagents, when it comes to input quality for research. And that is something that will require a rather radical response, including technology, such as automation, barcoding, reagent tracking, etc. To provide kind of one more example on what this looks like in the biomedical, in the biochemical, and in the kinase space in particular. As of now, IC50 curves will oftentimes look like this. There is a traditional serial dilution approach, where there is a curve with maybe 10 or 11 dose points, inhibitor and inhibition. Very, very simple curve, very simple graph. And that is the same approach kind of that we've been choosing. And this is kind of how we've been rethinking and redesigning this process, where instead of having a very small number of data points, we suddenly have a far greater depth of data points because we can print different doses with far greater resolution, thus increasing our confidence in the findings. And at the same time, what we can also capture is very extensive metadata, including, for example, equipment information, work slot information, run details, environmental parameters, quality control parameters, exact timings, all of those additional factors. So this is broadly referred to as metadata. And what this means is that suddenly, for one and the same assay, we're moving from an isolated curve that could have been generated under one out of many different conditions to something which is contextualized, which is well annotated, which provides real value to a researcher. Because suddenly, kind of with this new digital approach, we really have a data set that is also FAIR compliant. And of course, kind of the FAIR data movement is something which over the past 5 years has really seen increasing adoption worldwide, because there has been a realization within the research community that data has to be findable, accessible, interoperable, reusable, because otherwise it is not going to be kind of a good basis for research and for further progress. So I think this is kind of a very nice example from the biochemical and kinase space to show what this could look like. As a brief summary, kind of what I think is very important at this point to take away from the presentation is how really robotics and AI are hugely complementary. Because on the one hand, we have robotic assay execution, which means we do have the protocol adherence, we do have the reproducibility, we do have the full data capture, as well as all the verified reagents, cell lines, etc. So there is kind of that side of the equation that mainly refers to what comes out of our laboratories. And then there's the other side of the coin, which is what goes into the AI drug discovery programs. And that is suddenly data that is standardized, validated, mineable. And when taking those two together, kind of where we arrive at is this very interesting point where suddenly we move drug discovery to something where prospectively model training and model validation can almost happen in real time. At the moment, AI-driven drug discovery is still very much slowed down by the time it takes to validate models and by the time it takes to then do the next iteration. An AI model might suggest a new compound, but then it will take weeks, if not months, for synthesis, testing, data generation, quality control, to then start the next cycle. And that is something where I believe that not right now, but very, very soon, we're going to be in a situation where this process can be accelerated tremendously, where we can close this DMTA cycle, design, make, test, analyze, in a way that there is an almost instantaneous feedback loop between experimentation and data generation. Ideally, what we want to move towards is having a fully scalable R&D environment where the whole process from idea generation up until IND filing is becoming one continuous cycle of improved data, leading to better decisions, leading to faster project progression. What I would like to show kind of towards the end of the talk is what this looks like in practice. Because we've spoken a lot about conceptual background, we've spoken a lot about the data aspects and kind of the data aspects as they relate to principles such as FAIR data. This is now a very concrete example for a project we completed last year, where we combined our robotic laboratory in the UK together with, in this case, in silico medicine's AI capabilities, where they were generating new sets of compounds against the JAK family. So they were working on designing new JAK inhibitors because there was reason to believe that JAK inhibitors would hold promise in COVID-19 treatment. And what then happened was that while in silico medicine focused on the generative chemistry, Arctoris then came in to deal with the wet lab experimentation, with the profiling, with generating the data that would then basically feed back into the cycle. So this very much is kind of an example of something where the loop is getting very, very close to being a closed loop with the integration of AI and robotics. What that meant kind of on the kinase side was that thanks to automation, the whole process from acquiring the reagents via validation to assay characterization, optimization, profiling, and data processing was completed in less than 100 hours. Less than 100 hours for about 1 million data points. So that is kind of a power that only automation can provide, both in terms of speed, but again, also in terms of quality. To show what that looks like, we're going to share kind of a few more scientific graphs. So what we can see on the left-hand side is 4 graphs that show exemplary JAK inhibitor profiles. The 4 different colors, red, blue, purple, and green, those are the 4 members of the JAK family, JAK 1, 2, 3, and TYK2. So those are the family members. And we can see kind of how those 4 different exemplary inhibitors show an extremely interesting profile, whether, for example, selective for one, or whether partially selective for one, or whether they might kind of staggered response across the whole family. So it's very, very interesting, high resolution data that allows us to draw conclusions based on how these molecules have performed across kind of a very interesting therapeutic family. What we then see on the right-hand side is there's kind of one, basically is kind of the repeat. Is in this case kind of one inhibitor, how it's been performing against kind of those 4 different family members. We can then also see a time course where we looked at product formation rates over time. And the reason why this is so critical is because that allows us, kind of this time resolution, allows us to elucidate patterns in the data that a stationary one-time approach could not. What do I mean by that? If we now look at the two bottom graphs on the right-hand side, we can see on the left-hand side, there is an example for a kinase inhibitor that shows quick equilibration, but only a moderate potency. So in this case, with the inverse of the IC50 hovering around maybe 6.5. However, because equilibration happened very quickly, if the researcher was only taking, for example, a time point at 300 seconds or at 600 seconds, that would look like a potent inhibitor. On the right-hand side, we can see a high potency inhibitor, but one that shows a much slower rate of equilibration. So if we do the time course, we can see the green curve. We can see the green curve is growing over time, where, for example, at 1500 or 1800 seconds, we can see that inhibitor actually being more potent compared to the one on the left-hand side. So only by looking at the time course can we actually see how some of these inhibitors, while slower, are actually more potent. And that can provide extremely valuable information for the med chem team, because then the medicinal chemists can walk away and can think about how to optimize the structure further, either for potency or for equilibration speed. And kind of final remark is the red curve shows the Z prime. You can also see kind of how, again, thanks to automation, Z prime values are again in the territory that for a manual laboratory is very, very difficult to achieve. I'm going to leave it here. I'm just going to have kind of one final slide, basically to wrap up, I'm going to summarize some of the key points that I believe are quite critical to note. The one is that the future of drug discovery will be computational. We are going to see a further increase in the use of AI and machine learning and deep learning across the whole drug discovery and biomedical research spectrum. At the same time, robotics has become an extremely powerful tool, not just for high throughput screening, but for the entire discovery journey. And the reason why it's so transformative is because it works on both quantity and quality of data at the same time. Finally, we already see these technologies being deployed in silico medicine, in situ recursion, and others. So this is no longer something which is a theoretical exercise, but it is already being used and is already out there. So I think the next few years will indeed be extremely interesting and exciting when we can see more and more researchers benefiting from these very powerful technologies. And with that, I would like to finish the talk here, and thank you very much for your attention.
Thank you so much, dear Martin-Immanuel Bittner from Arctoris, United Kingdom. This was a true honor and pleasure to listen to your amazing talk. We learned so much from you about the important role of AI as well as the important role of robotics and drug discovery. Clearly, this was also a futuristic talk, which shows us a vision of where the drug discovery is moving forward. So thank you so much, Martin, for this amazing talk. Ladies and gentlemen, if you have any questions for my dear colleague, Martin-Immanuel Bittner, the floor is open. You're welcome to ask any questions you have. Just introduce yourself, unmute yourself, and go ahead and ask the question. Any questions, ladies and gentlemen?
May I ask a question?
Oh, please.
Yes, of course, Dr. Goldblum. Please go ahead.
I wonder if your with your robotics, did you look also at the effect of the molecules on the viral construct or some viral construct that is similar to SARS-2?
Yes, we did. It's a very good question. So when the COVID-19 pandemic started, we then very quickly established biochemical assays against the JAK family, which are involved in immunoresponse against the virus, but also against the main therapeutic targets for SARS-CoV-2, namely MPRO, PIL-PRO, and spike ACE2. So we also established assays against those, and we've had a lot of collaborators then use those assays to elucidate whether the molecules they're working on show response against one or maybe several of these targets.
May I ask if you found any repurposed molecules that could affect the virus or a viral construct, which is built with, such as a lentivirus with spike or?
So there were a few promising hits in the work that we did, which is currently still undergoing further study. I mean, most of these programs are not ours, but our partners, which means I can't really talk about them that much. But what I was so impressed with was to see how many researchers worldwide did change the research direction and did pool their efforts and focus their efforts towards repurposing of drugs and towards thinking about what would be potentially promising therapeutic agents because they worked in a similar viral class or because they worked against similarly designed receptors, etc. So what for me was great to see was seeing all of these people worldwide really come together and work on this very, very constructively. So that for me was the most impressive thing to see.
Well, thank you. It's a very interesting combination of the robotics together with the AI. I like it a lot. Yeah. Thank you.
Thank you very much.
Thank you, Dr. Goldblum, for the amazing question. And any other questions we have for Martin-Immanuel Bittner? The floor is open. If not, I'll move forward. So going once, going twice, gone. So